We will move to pillar 3. So we will move to Meelad, and we will have a series of questions and answers maybe at the end of his presentation.
Good morning. Can you see my presentation?
Yes.
Okay, in a full screen.
No, we cannot see it in full screen.
Okay. Yeah. And now?
Yes.
Yeah. Okay. Thank you very much for this interesting webinar to present our project to our colleagues and people from outside of the project. So, as you said, I will present the third and final pillar of the project, which called the FAW manage or control. And this pillar, we will have the effort to search eco-friendly method to manage FAW in Europe. I am Meelad Yousef from the University of Cordoba. We already learned, detected, and now we will manage or try to manage FAW before arriving or after arriving to contain its establishment or to control preventively. No, so during my presentation, I will try to only show you the main content of this pillar, beginning with the purpose of the pillar, approach of the pillar, and relevance, connection with others, especially how stakeholder can interact with the output of the project and benefit from this pillar. As you already also show that we have a lot or heavy intra and inter connection between pillars, between work packages, and between tasks also. So we will try to also to clarify these connections. Pillar 3 have two main purposes or main objectives. First of all, we will try to identify sustainable and environmentally friendly methods to control FAW in the European Union. This main objective or key objective of the pillar is composed of three work packages, 5, 6, and 7. In the work package 5, it's also key objective 5 of the project, we will search for natural enemies to be used under European Union conditions to control FAW. In the next one, we will search for microbial agents to also enhance this eco-friendly toolbox for protection against FAW. And also in the work package 7, we will go through cutting-edge plant-based and natural semiochemical solutions for FAW control. And the second main objective of this pillar is to integrate all these findings into work packages 5, 6, and 7, also from previous ones within a toolbox or indeed plant health guidelines to be accessible and practically used by third users. This will be done through the work package 8, entitled as Integrated Management Solution into Guidelines and Toolboxes for European end users. So let's go through these work packages and the objectives of each one. The first is to search for natural enemies and agroecological methods for FAW management. The first task of this work package, I should also indicate that the work package 5 will be led by Gabi. Work package 6 will be led by our colleagues from Greece. Benaki Phytopathological Institute. Work package 7 will be led by Instituto Canario de Investigaciones Agrarias, ICIA, and work package 8 will be led by our colleagues from Germany, from JKI. Sorry for I mean, it's difficult to read the whole name. So you can ask about the whole name. So the first task is to search. As a first step, we will search for native parasitoids and predators as macrobiological agents, as potential agents to control FAW. For example, we will begin not only searching parasitoids and predators for mainly for FAW, but also for other Spodopteras species. For example, here in Spain and also in France, we have a very good candidate like Hyposoter didymator. You can see in the picture is an autochthonous one and very good parasitoid for Lepidopteran pests. So we will search for to have a set of parasitoids and predators autochthonous. And then to evaluate this, the efficacy of these biological control agents and to select the best one to be used within augmentative biological control. Also, we will, and as a second task, we will study the conservation strategy of biological control. So we know what we have as autochthonous natural enemies. So let's see how we can manage as under conservation approach strategy to improve their efficacy. Also, after selecting best candidates, we will establish colonies and we'll try to use these natural enemies within an augmentative biological control strategy to release in at field conditions in, of course, in countries with the established like Greece and for example Canary Island in Spain or Israel. So we will try to study the best candidates at in an augmentative strategy at field conditions. Then very important also to include the entomopathogenic nematodes. Here we included nematodes within macrobiological control agents. Some scientists include nematodes within entomopathogenic or microorganism. Okay, so it's good to include also here. For nematodes or entomopathogenic nematodes, we will study through so selection of that are already identified, isolated in some countries. For example, our colleagues from INRAE have more than 50 nematode strain that they will study to select the best ones. But also we will search for natural occurrence of another strains of entomopathogenic nematodes. The study of these nematodes will include not only the efficacy but also to try to provide new formulations based on gel formulation to be applied also using some machinery like rovers to facilitate the application and to decrease the cost of application of these nematode by a new formulation. No? And the final task, and very important one, that our colleagues will try to also to search for exotic parasitoids, to try to establish at least a dossier with one or more parasitoids that will be suitable to be introduced to Europe by classical biological control. So we will search for non-native parasitoids. During the project will not be, I mean, you know, this type of programs is very, very long. So during the life of EUFAWREADY, maybe we will not reach to release a non-native parasitoid, but at least we will provide one dossier for the most suitable parasitoids, non-native parasitoids that maybe in the future could be introduced to Europe, and the effect of, for example, study agroecological conditions, if it's suitable in Europe to be introduced, etc. Also will be included in these work package. So these are the main tasks of this work package 5 for natural enemies and agroecological method. The next work package 6, in which we will have the effort to utilize microbial agents for management of Fuji beta of FAW in Europe. The first task is similar, for package 5, is to search and identify microbials against FAW. This will include the search for strains for entomopathogenic fungi, for example, bacteria or viruses that are entomopathogenic to FAW and to other Sporoptera species also. In this sense, for example, here at the University of Cordoba, we have a collection of more than 1,000 strains isolated from soil and phylloplain of Spain. And we will proceed with other colleagues from Greece, Israel, and others to study the efficacy of some strains to identify the best ones for the next step. So here we will search for microbials from collections already established, and also for new, to search for natural infected ones and to identify it if possible. As a next step or second task in this work package, we will study the complementarity between entomopathogenic nematodes and entomopathogenic fungal strains for the control of FAW in Europe. This is, I think, very interesting also approach that's we already began a collaboration with INRA to see if these entomopathogenic nematodes can be used like a vector or vehicle to transport entomopathogenic to transport the spores of entomopathogenic fungi in the habitat, in the soil. The spores of fungi is is like a passive movement, so they need a vehicle to be transported to, for example, soil dwelling stage of FAW. So nematodes can do this work. So we will study this complementarity, like compatibility and complementarity between these two microbial agents. As the next step or third task, the best strains of these microbials will be subjected to an improved formulations. Our colleagues from Israel is very expert in the formulations, for example, and they will provide an innovative formulations adapted to the application. Especially here we have a challenge always with microbials, the foliar application, because they are very susceptible to the abiotic conditions. So this task will be the main effort in this task is to provide formulation adapted to this foliar application. This will be done for the obviously for the most suitable or the most effective microbials from the previous tasks, not for all of collections of microbials. Another very interesting task in this work package is to go beyond the conventional use of microbials, like only for the direct control of pests. Here we will study mainly here at the University of Cordoba the innovative role of entomopathogenic fungi as plant resistance inducers. So we know that entomopathogenic fungi are microbes with multifunctional lifestyle, so they can colonize plant endophytically without any symptoms. They can induce plant resistance and control pests indirectly. They can promote also the plant growth. So we will use from our collection strains that we already know that they do this work in other crops like melon, like cucumber. So we will use this strain in maize plant, in the most common variety of maize. At least two varieties of maize will be included, and we will study how this fungi can colonize plant, can promote resistance, and also can promote plant growth and production. Okay, like it is another task. In the work package 7, we'll go, we will search for natural semiochemical solution for the management of FAW. This natural solution include the identification of novel semiochemicals, especially odors that can be used as repellent for FAW. So we will identify several semiochemicals and will be used also to, we will be tested to search the best ones and then to go with these best ones to the at least semi-field conditions to study their efficacy to be used as repellent for FAW at semi-field or field conditions. Another task in this work package is to identify bioactive compounds from plant and fruit byproducts. So our colleagues will go mainly by using, I think, tropical plants, papaya or another plants or products to search for bioactive compounds to be used also in FAW control. All these compounds, semiochemical or fruit byproducts, will be first identified chemically and then used at laboratory conditions and subjected to a selection process to, of course, go to the field with the best ones. Also, another task is to search green chemical products for FAW control. For example, we will study curcumin nanoformulations based biopesticides. So will be provided and their efficacy will be also tested at laboratory and field or semi-field conditions. After studying all these semiochemical or biopesticides, their effects on non-target organisms will be addressed within this task in an independent task because always these effects are important to be studied and showed for registration purposes in the future. So all the effects on non-target organism will be included in an independent task. In this work package, also as a last task, the improvement of native plant tolerance will be studied by the selection of the resistant varieties. So several varieties will be subjected to a resistant test, simple one, to several plants from several varieties will be exposed to Sporoptera frugiperda, and we will select the best or the most tolerant or resistant varieties to identify most interesting genes that can be used also in the future to develop new varieties or new resistant varieties for FAW management. So we will have a set of interesting genes here for resistance that can be used also in the future to develop new varieties. Okay, after identifying and studying and presenting the most promising micro, and plant products or semiochemicals, we will try in the work package 8 to integrate all these control method within a toolbox and guidelines for the end user. So the lessons learned from Europe, from other also regions, and from these, the output mainly from EUFAWREADY project will be integrated in practical and public and guidelines for the end user. So our idea in the work package is to develop an IPM toolbox for managing both established and transient FAW populations, also to provide FAW pest management decision guidelines to be used by the end user, in which we will include the most practical, economical, safe, and effective solution for FAW management. So yeah, as you can see, that we have a lot of crosstalk between the work packages and the tasks within each work package. I think it's a very important aspect of the project that this common work, this extensive crosstalk between the different pillars, work packages, and tasks within each work package to finally reach to this. I think it's this house, let's see, house represent clearly what EUFAWREADY want to present at the end. So we have a very solid base of learn and detect that is essential for any IPM program to know and to detect the monitoring, early detection, and then through all the pillar or objective that are a solid one to build the IPM toolbox at the end of the project. And thank you very much.
Thank you. Thank you, Meelad.